I think the way they're discovered has changed a lot because we have so much better a biological basis with the genomics and the understanding of the genetics of cancer. There are many, many more targets against which drugs can be designed. Also, the chemistry and the biochemistry, the methods for designing and making drugs to fit a particular target are far more sophisticated than they were. And in particular, if you take a kinase target, many people who have the resources can almost guarantee to produce a drug against a particular target. So the way that the drugs are discovered and made in the first place has changed. When we get to the clinical evaluation, that has changed a bit, but perhaps not as much as you might imagine. Because although we make targeted agents, they're not always targeted only to the cancer. So the biochemical target that's important for the cancer may also be important for some normal tissues, such as the skin. So we still have a problem of getting the right dose so we get an anti-cancer effect without too much toxicity. And that means that the phase 1 part of a trial, where you're trying to find the right dose, is still done in a very similar way with escalating doses. The difference is we also have more sophisticated ways of measuring whether the drug's having the right effect on a target as we go up the scale. Major successes in drug development for one individual aren't that common, and I've probably been lucky in that I've been involved in about 3 over the course of my career. First one was obviously carboplatin. The second one was the antifolate drugs that led eventually to the development of pemetrexed. But I think really the most interesting one has been the development of PARP inhibitors, because with the discovery of the PARP inhibitors' activity in genetic breast and ovarian cancers, the ones that are called BRCA tumours, there's really been a completely new paradigm for treating those patients. And of course the first PARP inhibitor, which is olaparib, has been licensed now. There's a bunch more in clinical development, I think, several of which will become licensed in the next year or two. And for quite a wide range of patients who have inherited cancers and who maybe at the moment don't even know that's why they've got cancer, these drugs will be very useful in improving their treatment. Nobody ever said it was easy to treat cancer, and of course one works on a lot of drugs that maybe don't fulfill the promise, don't do what you hoped they would do, and eventually are put aside and not used further. Sometimes they do work, but they don't work better than the ones you already had, and sometimes for some reason that you hadn't anticipated, they don't work at all. I think now with the better understanding of biology, with the genomics, with the more sophisticated target selection, we should get fewer disappointments. We should get more drugs that are doing what we hope. But there's still a lot of complexity in the way that cancer works that we don't understand, so the potential for failures is still there. I think the important things are going to be patient selection, because we will be designing a drug and we may know that it'll work on a subset of cancers. For example, the PARP inhibitors, we know they'll work on people who've got BRCA-related cancers and maybe not on all of the ones who haven't. For things like some of the kinase inhibitors, which work on specific mutations that are only found in cancer, there's no point in trialing them on patients who don't have those mutations. So there'll be more selection. There'll be more trials because we have more drugs. We have to find a way of streamlining the trials so that we can get results relatively quickly without too much expense in the number of patients treated at insufficient doses and so on. However, having said all that, the main principle of an anti-cancer drug trial, where you have to work out what dose you can give, and you're probably going to have to increase the dose to a dose that at least has some side effects in order to get a sufficient action on the tumour. That principle is still very commonly present. The drugs where you don't need to escalate the dose as much as possible are quite unusual. I think don't make any assumptions, observe things very carefully. Do increase the dose even if you think you may have got to where you need to be, even if just to make sure that you're giving enough. You do need to have biomarkers, some way of selecting the patients who may do well on your drug, and you need to have some kind of measurement to know your drug is doing what it was designed to do, like a pharmacodynamic endpoint. But it's important not to place too much reliance on that endpoint because the way you measure it may not be all that accurate. I think one of the major things for a successful drug development, which is very hard to achieve and is often still being missed, is to keep a multidisciplinary input all the way through the process. So the tendency to silo, we'll make the chemistry, we'll do the animal studies, we'll do the toxicology, we'll do the clinical study, and then people not talking to each other in between is quite common, and it leads to making bad decisions at every level. It isn't easy to make a drug for cancer. All the successful ones that I've been involved with have had lots of problems that had to be overcome. And all of those problems can be overcome if you've got the whole team talking together. And they can't be overcome if you have got everybody doing their own bit in isolation. So I think the collaborative and multidisciplinary approach is the most important thing.